This is episode 62 of the Immunology Podcast, T Cells and Solid Tumors with Dr. Kristin Anderson. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Kristin Anderson from the University of Virginia on the podcast to talk about her research on engineered immunotherapy for solid tumors. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
we'd like to remind our listeners about the upcoming International Union for Immunological Societies Congress taking place in Cape Town, South Africa, from November 27th to December 2nd. We are very excited because we will be attending, so you can visit iuis2023.org for more information.
Well, so we both had holidays recently. I had Labor Day, and apparently you were on holiday as well, right, Brenda?
Indeed. I mean, was August after all. You know, here people don't work. August is most people don't even work in this continent.
I know it. Believe me, for someone involved in transnational manufacturing, it's frustrating.
Yeah.
Did you go? Yeah. What country did you go to, Brenda, on your small little, drive or train?
I went to the beautiful mountain country of Switzerland. I visited the Mont Blanc, or Monte Bianco, like the Italians would say. It's really cool. There are still some glaciers there that haven't yet completely melted away. So that's pretty good. Pretty cool. Yeah.
Where I grew up in the summers, Mount Hood was where the U.S. ski team trained because you could ski on the glacier in the summer there.
Nice. Then you know glaciers.
I do. I have fallen down a couple of them.
Have you? How come you're still amongst us?
Because I eventually stopped falling. You can go 1,000 feet and then stop. And then your skis stop you. And then you get back up and keep going.
That doesn't sound safe at all. But well.
Skiing is not a safe sport, Brenda.
Yeah, but falling down like a glacier, that also doesn't sound. You can ski like normal people on like just regular mountains.
I would argue regular mountains have glaciers and that all the other mountains are just wanting to be a full-grown mountain with a glacier. But for my Labor Day, I painted little miniature figurines with my son, who's now getting into the BattleTech war game. For any of the audience who actually knows what that is, I have a 9-year-old who's going deep into war games all of a sudden. So that's been an interesting experience.
Wow, look at that little guy making his father proud.
Both proud and very busy, because now I have to read all the rules and then explain them to him, and he reads them too, and then we have to catch up and then have 8-hour battles.
Very nice. I wish I had a kid that would play, well, maybe not war games, but some other kind of game. That sounds kind of fun. Sounds like you're building yourself a little game companion.
Yes, we've progressed well past Settlers of Catan now.
That's pretty good. I mean, for a 9-year-old, like moving past Settlers of Catan, good parenting. I like it.
I'm trying. It's a skill. We're working on it. Dungeons and Dragons we've done some of, but we'll do more later. Does
it get hard? Because then kids, you need to, for Dungeons and Dragons, you need to have like some continuity, or like, does it take too much focus for a child to?
It's more that I have to also game master it and be ready, and I just can't keep up with how often he wants to play, which is now why I'm appreciating the notion of professional game masters that you can just hire, because like it's a lot.
Can't you just get like ChatGPT to like game master for you, just like forever and never tires and never always has something new to say?
That would be interesting. I know. I bet you someone's done that.
Yeah, for sure. I guarantee you there's an app that uses some AI, language learning model that makes these things. So I should then you don't even need to worry about it. Your kid can just play with the machine, and then what could possibly go wrong?
We're gonna have Skynet soon. That's all I can say.
All right, let's move to a brighter topic, because we're going downhill here.
We are. All right. So in terms of high computational needs, segue, let me talk to you about the paper High Resolution Landscape of an Antibiotic Binding Site in Nature. First author being Kevin B. Yang. Last authors are Aviram Rasuli and Evgeny Nudler. Nature, August 30th. It came out. So this gets into the molecular biology, biochemistry weeds of RNA polymerase in bacteria. So antibiotics often bind to parts of important enzymes for replication. And so rifampicin is an important antibiotic. It binds the RNA polymerase of E. coli, and they did a site-by-site mutation mapping using a process called MAGE, multiplex automated genome engineering, which they've produced many of the papers on, from what I can tell, in the field on using MAGE. I believe that is kind of their thing, or at least some of the authors' thing, to an extent, from what I can tell. But MAGE is an awesome name. First off, great acronym, love it. But what they do is they first did a MAGE profile of the specific binding region that's already known for the antibiotic, because you don't need to mutate everything. Then they looked at just what happens after that without antibiotic for selective pressure. Are they causing a selective effect? Yes, no on its own, and then use that as the baseline to compare to antibiotic addition, right? Because if you mutate it and has some other selective effect, you don't want to throw the antibiotic on top and just confound everything immediately. So they do the work. They identify there's this alpha helix that they go after, and they find some important residues. And if you're a structural biologist interested in RNA polymerase, you should read this paper for that reason. But for kind of the higher level interesting part, they find that things that cause rifaximin to bind tighter turn it from a bacterial static to a bacterial cidal antibiotic. So it's causing death. And then they do some really cool studies looking at sequencing of open genome. I don't want to say chromatin because they're not chromosomes, of open genome. And they show that what's happening is that in the mutation, the RNA polymerase is getting stuck. And so then when more so during replication, this doesn't happen at baseline, but during replication events, you're causing a traffic jam. So that the DNA polymerase coming behind the RNA polymerase runs into it, and then you have death and double-stranded breaks. And so they're saying they see that because eliminating Rec A or Rec BCD, which are the recombination proteins for double-stranded DNA, makes these even more bactericidal. And then they also see it just from some sequencing data. So that's the first thing that they found. And the second big discovery that they found was that things that make the RNA polymerase go faster, while it causes a fitness advantage at low temps, if you have something that depletes nucleotides, such as antimetabolites, it causes the antibiotic to work even better, because it causes the mutation actually on its own or with the antibiotic to work even better, because then what happens is, it doesn't cause that antibiotic to work better. It causes other antibiotics like sulfa, which is an antimetabolite effect, to work better, because they deplete the nucleotide pool so fast that then they starve before they're done because they're super speedy. So you can actually have mutations that cause super speed, which have a fitness advantage at baseline at a 35C growth. So that's good. But then if you starve them or inhibit them in any way, they fall off the wagon and die. So there you go. Some antibiotic structural biology.
I guess it's always good to know exactly how our antibiotics are interacting with our deadly pathogens. And this is, or which type of which antibiotic again are they studying?
It's rifampicin.
Rifampicin.
It's one of the ones I promise you've used to culture bacteria. It's like pretty common. It is. It's called an antimycobacterial, so it's not common for human infections. It's used for Mycobacterium avium is very common. It also works in E. coli. It helps protect against TB, so it's an important antibiotic, generally speaking.
I think that's where I heard it from. TB. That's where
it works on E. coli. It binds to RNA polymerase, but there's lots of mutations. And they also do some mutational analysis, by the way, look at other bacteria and see how it's conserved or not, and then find that some have that fast mutation at baseline and some other things. They do some evolutionary biology mapping as well.
And is there any way this study could help maybe make a variant of rifampicin that is more broadly applicable or that is resistant to some mutation or something like that?
I'm not sure they quite show that, but it does show where the changes in mutation are happening and the selection pressures on it for general antibiotic understanding. So the notion that some of these with this fast process, because that's all about the RNA polymerase, are more susceptible to sulfa. That's like oh, maybe you can identify that and know what species are better to go after.
Okay. Well, very interesting. I don't think about antibiotics enough. Sometimes I feel they're such.
So by 2050, it'll be the second leading cause of death after heart disease. It'll outpace cancer. It'll be antibiotic resistant infections will outpace cancer by 2050.
Great. Thanks for that.
Welcome to the 18th century again.
Awesome. That's exactly how I wanted to, transition to my paper. The doom is coming. There's nothing, all of this is irrelevant because we're all going to die of antibiotic resistant infection, which is probably true. Anyway. Okay. So on a lighter note, let me talk to you about gamma delta T cells and how a new study highlights a different angle of how gamma delta T cells can monitor cell health through their the ligands that they recognize on target cells. And I think it's very interesting. It's a really cool story. So the paper is called CRISPR Screens Decode Cancer Cell Pathways That Trigger Gamma Delta T Cell Detection. And the first authors are Murad Mamedov, Shane Vidova, and Jacob Freimer from the lab of Alexander Marcin at UCSF. And kind of long story short, so they use CRISPR screens to try to find genes or regulators of the recognition of a tumor cell line that they use by gamma delta T cells. And we know that gamma delta T cells, they have this special TCR that it's encoded by, it has this gamma and a gamma chain and a delta chain. And there's different subsets of gamma delta T cells. The most abundant is the one that has the the delta 2 chain. And particularly the gamma 9 delta 2 chains are one of the most abundant gamma delta types present in humans. And this is always kind of difficult to study because gamma delta subsets are quite specific to humans. They're not really comparable with the mouse version. So it's hard to also, I think that's why our understanding of gamma delta T cells is we really still not complete because it's not easy to study these cells. You need to study it in a human setting, kind of. So they focus on these gamma 9 delta 2 T cells, which have this known specificity for a complex composed of BTN3, BTN2A1, BTN3A1, BTN3A2, and in which, BTN stands for butyrophilin 2, butyrophilin 3. Butyrophilin is a ligand that is the known ligand for these particular gamma delta chain, and they associate together on the surface. And they respond, so they are expressed on the surface, responding to the presence of certain metabolites inside the cell, certain phosphometabolites that are derived from a metabolic pathway, the mevalonate pathway. And these metabolites are both of bacterial origin, and I think initially a lot of the focus was on the possibility of these phosphometabolites to be sourced from bacteria, and therefore indicators of bacterial intracellular infection, and therefore that would be the kind of usefulness of these gamma delta T cells that are indirectly sensing these metabolites. But we also know that in particular, tumor cells that have a dysregulated metabolism also have an increased abundance of this phosphometabolites that are basically kind of phosphoantigens. And that we also can see that gamma delta T cells of this particular subset can recognize under certain circumstances tumor cells and kill them. So they focus on this interaction, and they do a CRISPR screen looking at different genes that not only are affecting the kind of the two factors that will modulate the recognition by gamma delta T cells, which is the expression of phosphoantigens, but also the surface expression of the BTN protein itself. And kind of they do a lot of they show they have a really nice platform to do this, and I'm going to kind of jump a little bit to the conclusions because I think it's very interesting. They find according to the kind of genes that they find, they realize that it's not only the abundance of phosphoantigens that is regulated inside the dysregulated metabolism of the tumor cell because of the metabolic pathway itself that is increased, but also there's direct influence by certain metabolic sensors that will modify the expression of the BTN proteins themselves. And I think really the most interesting result that they see is that there's one particular protein which is AMPK, which is a very well-known regulator of cell metabolism, and it is activated upon it can sense a lack of ATP. It's activated it's called AMP. It's activated by high amounts of AMP in the cell, which signal a low energy situation, and usually it's activated in tumor cells because of this metabolic dysregulation. And so they show that one AMPK activation actually can upregulate BTN3A, in particular this component of this complex, therefore providing a more or less direct way in which the internal metabolism cues that are reflecting the metabolic state of the cell can also directly influence the expression of this complex on the surface, and therefore make the cells more susceptible to gamma delta V, so gamma 9 delta 2 recognition and lysis. And I think this is really interesting because then it kind of adds a different layer of activation, of different layer of regulation of how these gamma deltas can recognize the tumors. And they show that they do this both by metabolic regulation of the mevalonate pathway itself, but also through expression of these particular the complexes. So I think it also adds more credence to the idea that gamma 9 delta 2 cells can also be important tumor control cells, but because I think they're usually more associated with bacterial infections and the metabolites that come from intracellular bacterial infections.
I start wondering how much when we hear about tumor microenvironment, it's all mediated by metabolites because of tumor's weird metabolism of everything.
Yeah.
But, you just gave me the world's best segue.
I'm
going to talk to you about immunometabolism.
Awesome.
I'm going to talk to you about metabolic programs of T cell tissue residency empower tumor immunity. This is Nature, 30th of August, 2023 here. First author is Miguel Reyna Campos. Last author is Ananda Goldrath. And this paper goes pretty deep, but I'm going to try to keep the summary at a high level here. So tissue resident memory CD8 T cells, so TRMs, as this paper calls it. They come on rapidly. They offer long-term protection. This affects tumors. It affects infection. They do an immunometabolism sweep, knocking out CRISPR in mice to start with, T cells that then convert to resident memory T cells. So they kind of start this by taking CD8 T cells, doing this, lymphocytic choriomeningitis virus, this LCMV way of tracking viral response, do various knockouts. They use the P14 T cell receptor transgenic system with the specific glycoprotein. So they're inducing a response. And then they basically set up the system with a screen on top of it for metabolic genes and come in on the cholesterol pathway as being really important for establishing tissue residency, but most important in the small intestine. And then as you get more and more robust in knockouts, so CRISPR-Cas9 being the greatest, but they then verified this with hairpin RNA or a siRNA versus a full induction of shRNA by genetic induction versus full CRISPR. So the bigger the dose of knockout of this pathway, and I'll get the specific genes in a second, but they show dose-dependent effect for all of this, where it's most important in the small intestine and less in other things, but there's some tissue specificity to how much it matters. So the first gene they really come after is SREBP2. This is a sterile binding gene. It senses the sterile regulatory element binding protein 2, senses cholesterol, leads to downstream signaling, right? And has a negative then regulator of cholesterol synthesis, actually. And if you, it's really important in tissue resident cells, especially in the small intestine. You knock it out, you get less memory cells. So that's their first finding. But then they did some cholesterol signaling and noticed that it wasn't really cholesterol dependent. And in fact, if high levels of cholesterol downregulate SREB2 from negative inhibition, right? Feedback. And so it actually doesn't help. What they find, though, is by looking at some other genes involved and looking at effects of statin and everything, it definitely is statin dependent. So that's internal synthesis of HMG-CoA reductase. That's what the statins block. But the cholesterol pathway, it's really the mevalonate, which you had talked about, cholesterol pathway. And that has non-steroid. Remember, cholesterol is technically a steroid or a sterol. Sterol, but it's non-sterol components of that pathway and shunts. And they show that the shunting pathway to coenzyme Q, which is one of part of the electron transport chain, is vital. And that's where this all action is happening. So you do some nice knockouts. They do a knockout of a gene called FDFT1 that creates a protein that's responsible for the shunt. So if you knock it out, you have more shunting, essentially. It forces a shunt with knockout, so you get more coenzyme Q, and that leads to an accumulation in the liver of resident cells. So they do some more work. They do they do small molecule inhibitors and, again, dose-dependent knockdowns, and show that this coenzyme Q is really what's driving this. And so what happens then is that this enzyme supports mitochondrial respiration. They look at that pathway too. They measure mitochondrial respiration. If you have more mitochondrial respiration or you inhibit it, you get the opposite effect. You get more tissue resident memory cells. And then they show that the axis can boost tumor immunity. By boosting this axis, you get better tumor immunity. And then they even indeed look in tumors and can find that they did some human tumor cells and did some tumor models that show that this could lead to better control of the tumor, right? And I think they even went back into humans. It's upregulated in exhausted proliferating. It's actually upregulated in cancer and tissue resident memory cells. So to even get them into cancers that's been resected, you have to have this pathway activated. So it's coenzyme Q through the cholesterol pathway is a fundamental metabolite to generate resident memory T cells.
I guess that's in line with, we know that they have a very active mitochondrial respiration, and that's
Right.
what coenzyme Q is part of. So, is this surprising? What? How do they? I don't
Basically, they
know.
get it all the way down to it's this pathway, these molecules that have to do it. And if you inhibit it or supplement it in the right ways, you have downstream, mouse level clinical effects.
Okay.
They can do they did a melanoma model and then enhance the pathway and made melanoma go away better.
Okay. They enhance the pathways through overexpression or some small
They
molecules.
did small molecule inhibitions to cause the shunt, and then they overexpressed it as well. They did it multiple ways.
Okay. Yeah, I guess in it always, when it comes to this, metabolic pathways in T cells, they always does seem to look like healthy mitochondria is critical for especially the tissue resident cells to survive.
Yeah.
All right. Okay, so for the last paper of today, talking about survival, I have another also cool story. I like it. I think it's a clever idea. So this this paper is called um, epitope base editing CD45 in hematopoietic cells enables universal blood cancer immunotherapy. I thought it was pretty cool. So, first author Niels Velhausen, and from last authors Carl June and Sar Gill. Carl June, of course, at UPenn, and the same for Sar Gill. And yeah, whoop whoop! Here you didn't see, but Jason always gets happy when he hears UPenn.
Gotta love Penn.
Yeah. What was your mascot again? What was the go? Do you have a no? Okay.
I don't remember. I was a postdoc there, but Carl June,
Yeah.
that's episode 4, I think.
Yeah, he was one of the early. So thank you very much, Carl, for having believed in us early on.
He's a pioneer in all things.
For sure. Okay, so I think the title kind of gives the secret away quickly, but in this paper, the authors are trying to make a point that, of course, hematological malignancies are very diverse, and we have been able to successfully treat some of them using CAR T cell treatments. And this is because for some of the malignancies, there are very specific markers that we can target that can work, so that if you have a CAR T cell that recognizes a specific marker and gets rid of the tumor cell, for example, CD19, you can have actually a very for many patients, this can be a very effective treatment. Sometimes it does happen that if this marker, in the case of CD19, is not uh, indispensable for the proliferation or the function of the cell, it might be that cells can be pressured into losing it, and then you have a relapse, and that's also not good. On the other hand, different types of hematological malignancies will have different markers. So, CD19 will only be valid for malignancies that are like a B cell derived. Of course, if you have a T cell lymphoma, then that's not going to work, and that makes it harder for T cells. Which marker can you use that is not going to affect your therapy, that is manageable, because you can get rid of all of a patient's B cells, and treat them with immunoglobulins and they survive, but that's not the case for other kinds of immune cells. So what they look at is like, okay, can we find a marker that is widely expressed on any hematological cell, and that is can be kind of we can lose. And so they go around this point a little bit, and they kind of zero down on CD45. And CD45 is, as you know, expressed on all hematopoietic cells. So it's very wide. It's a very, very widely applicable marker. So you could basically target any hematological tumor with it. And so they they use kind of known uh, antibodies against CD45, and they end up zeroing in a couple of CAR constructs using these their variable domains, and they kind of compare them to see which of them is binding, and they find a couple of CAR constructs that would could potentially target CD45. And then they say, well, if we have a CAR T cell that has a CD45 specificity, of course, to prevent fratricide, we should get rid of the CD45 in the CAR, in the CAR T cell. And this is where, of course, if you think about it, the CD45 is a phosphatase that is important, particularly, in the case of T cell signaling, but also for the development of immune cells in general. So actually getting rid of CD45 is not, at the long term, is not great. They show that you can have some initial response, but then eventually cells stop functioning, and then you have a relapse. So it's not a durable solution. So what they do, and I think it's really cool, is they say, well, we cannot get rid of CD45, but can we get rid of the epitope that is recognized by our favorite CAR? And that is exactly what they do. They use base editing, and they're lucky enough that they find, so base editing with Cas9. In this case, they have base editing with Cas9 functions that you have the kind of the domain that binds the DNA of Cas9 intact. So Cas9 can find the target sequence, but the cutting domains are deactivated. So instead, what you have is a deaminase bound physically to Cas9, and then it will change some of the nucleotides in the region where Cas9 is binding. And with some luck, you will have that the mutation that it generates will either change the sequence of your reading of your coding sequence, or make a change that will result in a modification of your final protein. And that's exactly, so they do try a little bit. They find some targets, so they need to find out first where are the antibodies recognizing, which part of the CD45 protein they're recognizing, and then try to target that area with this base editors, and hope that they will find a dominant mutation that generates a change in the epitope that is sufficient to prevent recognition. And they do this. So after I kind of imagine took some work, and they do that, and they show that this edited version allows them not only to change the CD45 sequence of the CAR T cells themselves to prevent fratricide, but then you can even combine this with a bone marrow transplantation, in which you base edit the hematopoietic stem cells before, and then you end up having, at least depending on the efficiency of your editing, then you end up having at least some of these cells with this modified CD45 sequence that will then not be susceptible to the CAR. And then you could potentially use this as a kind of combined therapy, and you could use this for any kind of hematological malignancy. They also show that you can even do this. You don't necessarily have to have a CAR. You can also, if you do this transplantation of edited stem cells, and then you have, for example, a bispecific activator. So that, for example, the CD45 antibody bound to a anti-CD3 antibody, then you can activate cells. This would also work, and really differentiates endogenous or your original hematopoietic compartment, where that includes your tumor, from the new stuff that is immune to the detection by this particular clone. So I thought it was a really very clever. I liked it, and it makes me think, can I use this idea in my research? And so I thought it was very, very cool. So nice work.
So it sounds like you can make, by tweaking this, if you had a pan CD45 CAR, it wouldn't kill itself. That's good.
Mm-hmm.
But then they're suggesting you do gene editing of the host as well, at least some of their cells, so that you don't kill all their immune cells.
Yeah, or if this person is going to get bone marrow transplant, then that bone marrow before transplantation, you edit it, and then you give this bone marrow to the patient. So the new bone marrow is immune to the activity of the CAR. And I think what's also important that I didn't clarify is that they do a lot of tests and they show that this loss of this particular epitope does not affect the functionality of CD45 at all. So that's also, at least in their models. So that's also very important, right? Because if you modify it, if you change an amino acid that is important for the function, then it's not gonna work, or it's not gonna be ideal. But yeah, the idea is that if you want to make, usually these people do get bone marrow transplants anyway, so you might as well, protect the bone marrow transplant, give the CARs, and then in principle, ideally, this would kind of fix the incompatibility.
Makes sense. All right, cool. That's neat. All right. Well, we've gone through CAR cell. We had an all T cell, basically, besides one bacteria Roundup today.
That is true. Yeah. I'm sorry. Just these are so interesting sometimes.
And we're going to be talking to Kristin Anderson at the University of Virginia in just a moment about T cells as well. But before we get to that, are you looking to improve your T cell therapy development, Brenda, in particular?
Always.
Well, with STEMCELL Technologies' new GMP manufactured Immunocolt XF, you can achieve robust T cell expression with high yield results and high viability without the use of serum. To learn more, visit www.stemcell.com/immunocoltxf. And remember, GMP manufacturing, which is awesome.
So today for our interview, we will be talking to Dr. Kristin Anderson. She is an assistant professor at the University of Virginia, and much of her work, I'm very excited about it, is about T-cell immunotherapies and targeting a particular set of cancers that have a particular association with her life. So I think we are going to have such a nice conversation today. Kristin, thank you so much for joining us. Welcome to the Immunology Podcast.
Thank you so much for the invitation. It's really an honor to be here.
Thank you for being here. All right, Brenda, this is your cell therapy land. So you let me go first with gut and microbiome. So fire away.
That is true. That is true. So, so as I very briefly mentioned, you have some very interesting work around using T cells for immunotherapy, focusing mostly on ovarian cancer. I think it would be really nice for our listeners to have an idea of what is your lab, what's your current focus in your lab, and what brought you to study ovarian cancer and I would say similar cancers. I think there's some personal story that I thought was very inspiring.
Well, thank you. Yeah. So like you said, I just started my brand new independent research lab at the University of Virginia. And the focus of the lab really is developing engineered T cell therapies that can overcome immunosuppressive obstacles in tumors. So I come from a lab where I learned how to engineer T cells to have a new T cell receptor. And that was absolutely incredible training that really taught me a lot about using physiologically relevant mouse models of disease, developing relevant human models of disease, and a lot of the really amazing molecular biology tools that we can use now to identify an obstacle and then engineer around it in the lab and build tools to bring back to the clinic.
So speaking of tools, you work a lot on OB-GYN tumors, so breast cancer, ovarian. How are the tools for that as tools in mice go in relation to human disease?
Yeah, so we are using mouse models that as much as possible mimic the physiologically relevant components of tumors. So we try to find immune competent mouse models, and we work in syngeneic systems. So every time we're doing engineering experiments, we have a fully mouse system: mouse tumor antigens, mouse immune cells. So the whole system, we, as much as possible, want to try to simulate what's happening in patient tumors. So every time we identify an obstacle in human disease, such as a ligand that might turn a T cell off, or a death molecule, or a suppressive cytokine, or a suppressive metabolite, we go back to those immunocompetent mouse models and really vet whether or not that component is also present, and is it present at similar levels. If all of those things line up, Then we work in the mouse model to try to engineer a way around that obstacle. And once we find one we think has potential to work, then we start doing the preclinical translation back into human models.
So that I think of course that is a very interesting approach. I mean, very makes a lot of sense that but I would always think, what is the risk of translation there? Like, what do you think are the major uh, steps when you find something that works in mouse? You might find a system that you can characterize, that you work with, but then what are the chances that the human situation will, transfer?
You're absolutely right. And this has actually been one of the obstacles in ovarian cancer, for example. So lots of immunotherapy concepts and tools have actually been tried in clinical trials. And to date, most of them have been fairly modest or even really disappointing in terms of outcomes. So what we've been really trying to do is build human models. So we have an intermediate step between that mouse and an actual clinical trial. So I worked with two collaborators at the University of Washington, Dr. Elizabeth Swisher and Dr. Venu Pilar-Setti, and we together developed this slice culture model where when a patient goes in for surgery, we actually get a live tumor specimen. We can bring it back to the lab and slice thick sections on the vibrotome. We can actually keep those alive in culture for at least 7 days. And that gives us an immune competent setting that actually retains a lot of the physiological relevant components of the whole tumor, that we can then have that intermediate model to test things in before we go to that next step.
That is, I was also familiar with similar model in which you analyze like little pieces of tumor, and then, for example, you look into the effect of checkpoint inhibition on a small scale, and that actually seems to translate very well with the response in patients. So I guess probably your system, you've tried it extensively, can also help you, for example, predict whether a patient responds to an immunotherapy or something in that level.
We're very hopeful. I actually just wrote a grant asking for funds to see if it is predictive. So keep your fingers crossed.
Okay, fingers crossed. Jason, cross your fingers.
All right. They're crossed. No one can see. So in breast and ovarian, and maybe the answer is different, what are the biggest challenges there for, cellular-based therapies? And I ask specifically because when I think about this, cell therapies work pretty dang well in what we call liquid tumors, so hem-onc tumors, and not well in solid. But I think of solid tumor, breast cancer, sure. But ovarian cancer is weird because it gets into the perineum and almost, in my mind, behaves kind of like a liquid tumor in a lot of ways. And so I was wondering if, like, for instance, does it T-cell therapy based, these therapies work better in ovarian cancer because of that liquid-like nature or anything like that?
Yeah. So that's a really great question. So the big major differences between liquid tumors and solid tumors, as you were alluding to before, is that really established microenvironment. And you've had guests on the podcast in the past who've talked about this. Like you recently interviewed Greg Delgoff, and he talked about the microenvironment that's really established metabolically. So we're dealing with issues like that. Ovarian tumors, you're right, are very unique in that they often, once the primary tumor seeds metastases, they basically form nodules throughout the peritoneal cavity, all over the omentum and other peritoneal located organs. And in addition to that, many patients develop ascites, which is a very liquid component that has tumor cells, immune cells, blood cells, all sorts of things floating around in it. So a lot of the work we do in the lab really focuses on that solid tumor nodule, with the idea that if we can eradicate that component using immunotherapy, there are tools already in the clinic to address components of ascites. Like there are protocols for actually draining ascites from patients, which can dramatically minimize the amount of tumor in that capacity. But yes, the hypothesis would be that if we can address the solid tumor component, which has a lot of obstacles we need to address, that the engineered T cells that can interact with cells in ascites, what is there, might actually be a little more effective than killing cells in solid tumors.
paracentesis is my favorite medical procedure to perform, which is draining ascites. Nothing is more satisfying than taking 5 liters off of someone's belly in a day.
Absolutely.
You also get it from liver failure. So you're never in a good spot when you have it, but it definitely they feel like way better immediately. And it's kind of crazy to see.
Absolutely. Now, you had asked before a little bit about differences between breast and ovarian cancer and some of the tools that we have. And so there are lots of new chemotherapies coming out. So some really incredible work has been done in the breast cancer space to identify not only chemotherapies, but some other agents that can be very targeted in terms of killing tumor cells. So one of the components that actually caused me to pivot in my research from being straight immunology, working on infectious disease and vaccines, into immunotherapy was my own cancer diagnosis. So when I was in graduate school, I actually went through diagnosis and treatment for triple negative breast cancer. And so I got to learn a lot very quickly about where cancer and cancer therapies were at that point. And so when I was going through treatment, I had a cocktail of drugs that were both general. So all breast cancer patients got some of these drugs, but I also had one chemotherapy agent that was targeted because I had a BRCA1 mutation. And so lots of really incredible research over many decades has gone into identifying if we can figure out specific things a tumor expresses that are targetable, let's develop a cocktail of agents that can really have high potential to eradicating someone's disease. And in my case, I was incredibly fortunate that that chemotherapy cocktail that they came up with for me eliminated my tumor before we went in to do surgery. So I am one of those cancer patients who is immensely grateful to the physicians and the scientists and the researchers who worked incredibly hard to discover and create those therapies, because I'm here today because of those. So that made me pivot into immunotherapy because I could really leverage my immunology expertise and pivot into an area that I'm really passionate about. So breast cancers, there are definitely people working on developing immunotherapies for breast cancer. Because there are even fewer therapeutics that are that effective for ovarian cancer, that was really the reason one of the main reasons I picked to be in this space.
So it's always so encouraging or so inspiring to find scientists that have such a personal relationship with their research. Of course, we're so glad that you're here with us and that it all worked out. And I think it must be very satisfactory to work on. Or not, maybe because you think it's never fast enough, you never get enough done. But I can imagine that having such a personal connection to your research must be something else.
It's absolutely incredibly motivating. I'm sure all the scientists listening to this recognize that there are days where you have put your heart and soul into designing and planning and executing an experiment, and then something didn't work, and you were just so frustrated. And I think the mindset that that's how science works, right? A lot of the time, we just have to keep improving things until something works and we can move it forward. That attitude helps. But the piece you're talking about, the motivation to build therapies to help people, I imagine a lot of scientists have. But yes, the fact that it's very personal for me, I think helps keep me optimistic and helps keep me pushing, right? Coming into lab on days where I'd rather stay in bed and read or, right, do something like go on a hike. I am more likely to get out of bed on a hard day and come into lab because I have that personal connection.
Yeah. So one more, like diving again into the research, because I really like T-cell therapies. I work on that, and so I'm always very curious about details sometimes.
Yeah.
So thinking of, ovarian tumors, for example, and looking if we were to find cell therapies to treat advanced ovarian tumors, for example, where are we in terms of which antigens can we target? Where do we understand? Can we use? Are there any CARs that can we use? Any TCRs that we have identified? I know that you have worked with mesothelioma as a target. Is that Like, is that an interesting target? Where are we? What is the future of cell therapy when you think about, for example, ovarian tumors?
Absolutely. So there are a lot of groups that are working on identifying really great targets. So Kunle Odunsi's group has some TCRs for NY-ESO. Dan Powell's group is working on some CAR T cells. Carl June's group is working on CAR T cells. So there are lots of antigens people are starting to identify and build tools for. And some of those are in early phase clinical trials. In my work, we use a tool that was already established in the Greenberg lab. So in Phil Greenberg's lab, when I joined, Dr. Ingen Stromness and Tom Schmidt had already developed TCRs for mesothelin, both mouse and human versions. So it was a really great tool for me to leverage because mesothelin is overexpressed in ovarian cancer. But the real focus of my research is a little less about antigen discovery and a lot more about, once we've figured out what antigens to go after, how do we further engineer these cells so they overcome the suppressive signals they're going to encounter in that solid ovarian tumor? So a lot of the tools that we build actually would synergize beautifully with any CAR or any TCR that we identify that is productive against ovarian cancer. And when we talk about antigen targeting, I think anybody in the field would agree with the statement that most of these tumors are so heterogeneous that targeting a single antigen may not be sufficient to completely eradicate, especially advanced stage disease. So I think there are lots of groups looking at trying to identify different antigens so that we can target multiple, either using cool CAR T-cell engineering strategies that target more than one antigen simultaneously, or making a cocktail of engineered cells that have different receptors that target multiple different antigens.
So, getting into your research a little bit, you've made the much lauded and often asked about transition from postdoc to faculty pretty recently. We were talking ahead of the show on your new office. So, I'm sure like 99% of people want to corner you and ask you how you did that and what's it like and all of that. So could you speak a little bit to the job hunt? I know you had a longer postdoc. You had somewhat of a shift from straight immunology to cancer as a postdoc, but those go together, right? You weren't going from like meteorology to cancer or something, right? It was in the same universe. But how was the job hunt? What was it like? Any words of wisdom for aspiring, postdocs or whatnot who are getting ready to try to make that jump?
Yeah. So as you said, my career trajectory was an experience. I inadvertently, unknowingly submitted my faculty job application packets in fall of 2019. So I had faculty interviews lined up for beginning 2020, and we all know that everything became chaos beginning of 2020. So most of my faculty job search got completely delayed by a year simply because everything shut down. And then when I was recontacted to move forward with all those applications, everything was virtual. And so I did virtual presentations, virtual chalk talks, lots of different interview formats to do first interviews. And then after vaccines were available, that was when my partner and I started doing second interviews where we would actually visit locations and look at lab space and things like that. The pieces of advice that were given to me that rang true, that were incredibly helpful, were be prepared. So know who you're going to talk to. I felt like the best conversations I had were ones where the person came in and said something about knowing who I was. It raised my spirits and made me feel so valuable and great. And so doing that for someone else, I imagine has the same feeling the other direction. So be prepared, know who you're talking to. That way, when you get into those conversations, you don't feel caught off guard or in the dark. And then when I was preparing my chalk talk, I got a lot of feedback on structure. So I saw other people's chalk talks. I read a lot of blogs about what people did. And in putting my chalk talk together, I was very I tried to focus on clarity. So minimalistic on the board in terms of words. And I really focused on bullet points and cartoon models. And that was actually something I learned from Mark Jenkins when I was in graduate school. One of the things that we were taught ad nauseum was you need to be able to draw a cartoon model because that's going to help people understand that you know what you're talking about. And we used to go to immunology retreats where we were allowed 3 slides. One was your title slide, one was your key piece of data, and one was your cartoon model. And that was all you had for the poster session to talk to someone about. And it led to some of the most engaging immunology conversations that I had about my work and much more high-level thinking. So I wasn't in the weeds about details of an experiment. I was much higher level, big picture thinking about the whole situation. And people could talk about my science even if they weren't experts in my science. And that really helped in my chalk talk.
So maybe a question raised, just, I don't know if I'm allowed to ask this, but how many places did you apply for? And how many, like, what is the kind of um, invitation rate people you should expect? And how was your experience? If you're, if somebody that's in this position, I mean, it's always so, you always hear, yeah, rejection is common. You don't need to get, don't put down, don't give up just yet. But is that was also the experience for you? I mean, also despite the fact that it was the middle of pandemic, so that might change the whole environment completely. But what would you think about that? The success rates that.
Yep, that's a great question. And I think it will differ depending on the specific focus of your work. So I had an additional component that I was trying to navigate during my faculty search, which is that my partner's also an immunologist. And he also has a research lab, and he's an associate professor. And so we are, I know a lot of people call it the 2-body problem. During the search, people kept telling me to think about it like a 2-body opportunity, because lots of people would want a T-cell immunologist and a B-cell immunologist. And so in some applications, I would read the description of what people were looking for, and it would only sort of peripherally talk about cancer immunotherapy. Like there might have been something else those positions were looking for. I applied to anything that sounded relevant, like they might be looking for someone with my skills. And I got contacted by a lot of places. Probably the majority of places I applied, I had at least a first interview, and most of them I had a second interview. Usually the thing that kind of dictated if I had a first to second interview transition was if the institution had the ability to support 2 scientists. And I think one of the reasons that my rate of getting interviews for all the applications I submitted had to do with the field that I am in. T-cell engineering, cancer immunotherapy is a really hot field right now, and especially translational immunologists who are building therapeutically relevant tools. One of the things I learned in my job search was that there are so many institutions that are just so hungry for immunologists who can work in that basic discovery space and teach graduate students and have postdocs, but who also can interface and collaborate really effectively with clinical colleagues. So many of the interviews I had actually ended up, maybe I initially was applying for a basic department, but they ended up trying to build a position where I was jointly appointed in the gyn-onc or OBGYN clinical department to better facilitate the research that I was proposing to do. And so I think, if you are an immunologist going from graduate school to your postdoc and trying to think long term, explore everything, find a topic that you are actually passionate about, because if you want your topic to be something you carry on to your independent research lab, it's got to be something you really love. But I also put a lot of thought into what is going to be a niche area for me. How am I going to make my research program something where there aren't very many of me or people like me? And so institutions might have a higher desire to have someone with my skills and my expertise.
So kind of a two-parter. Do you think you could be too niche though, so that like, oh, you're the only one and we want a more broad thing? But then other question, what did your funding look like when you were able to land a faculty position? Did you have Ks? Did you already have an R? Did you have nothing? Because I know in the US that very variable what type of funding you have and where you can get offers.
Please clarify for the non-US people what it means to have a K, what it means to have an R.
A K is a junior faculty level grant that supports your salary and a wee bit of funding for either reagents or a technician, but it's like your transition grant after a postdoc fellowship. And R is your main funding for a lab, so to speak.
So I can speak to both of those. So the first one was about too niche. So this was actually very deliberate. So what I was trying to do was find an area of interest where there weren't a lot of other people. So I wouldn't be necessarily competing with someone at the institution I was applying with, but the skill set that I was bringing was broadly applicable. So my research specifically, we do ovarian cancer research. But like I mentioned a little earlier, the tools we build would synergize beautifully with anyone working on TCR or CAR-engineered T cells, or even tumor infiltrating lymphocyte therapies. So there's a lot of opportunity for collaboration. And some of the obstacles that I'm addressing in ovarian cancer are present in many other solid tumors, pancreas cancer, lung cancer. And so if we find something in one disease that's relevant and we have collaborators who can help us test it in other models, we might be able to build tools that are therapeutically quite broadly applicable. So my research focus specifically is niche, but my skill set's very broad. So that was by design, very deliberate. And then the other question was about funding that I had coming into this. So when I first submitted my applications in 2019, I did not have any additional funding to bring with me. I had actually been really successful at getting grants as a postdoc. So at that point in my career, I had gotten around $750,000, $800,000 worth of funding just as a postdoc to support my postdoc research. And so that already I had a track record of being able to write grants and get funding from lots of different sources. But when the pandemic hit and we were stuck at home, that was when I decided I would write a K award. And so the K grant that I focused on was something called a K-22. Here in the US, we talk a lot about the K99 R00 grant process, but I was too far along in my postdoc to apply for that. The K-22 award is really designed for people in the position I was. You are so far along in your postdoc, you don't really need more postdoc training, but you would really benefit from mentorship as junior faculty. So that was the one I applied for. And I was fortunate enough to get one from the NCI. So when I got my first round of job offers, I didn't have a K-22. I had applied for it, but didn't know that I was going to be getting it or not. So it didn't really impact the offers I got early on. Once I was awarded it, it impacted things like salary negotiations and space, and some of those other components.
That's very helpful information. Thanks for sharing that. I'm sure many of our listeners were taking notes. And it's also so refreshing to have such a clear account of your experience. It's such a difficult transition really from the, especially and as a fifth year postdoc myself, at some point you start feeling the heat, and it gets here in Europe as well. So it's always nice to hear the experiences of people that are at least making it through. And so at the moment, what does your lab look for? So you've been like around 5 months in. So how big is your lab now?
Right. So I started officially at the end of April. And because I had a longer postdoc than most, so I had about 6 years as a postdoc, 6 years as a postdoc. And then I transitioned into a research associate position. Some places call it an instructor position. So it was essentially that transition position where I was still in my mentor's lab, but I was really trying to build the things I was going to bring with me for my independent research program. And so in that time, I did a lot of single cell RNA sequencing experiments where I obtained the data. So we did some pilot experiments, we really optimized design, and then we got a huge influx of data. And I am not a bioinformatics expert. I don't code. I don't do any of that analysis. So I was going to desperately need a collaborator or people in my lab who knew that. So as soon as I was able to start hiring, there were a couple types of positions that I targeted. One, lab manager. So hired someone to help me get all the protocols and paperwork done. Hired someone who could help me pick out lab equipment, get things ordered, and someone who could be on the ground here in Virginia to receive things while I was still in Seattle and my kids were finishing up the school year. So lab manager, we lucked into finding an absolutely excellent person who's part of our lab. So I'm really grateful that she was looking for work the same time we were looking to hire. And then I actually hired 2 bioinformatics experts. So we have one individual in the lab who basically did graduate school and a postdoc in bioinformatics and spent some time in biotech, and then came back to academia because he's interested in pursuing an academic job. So he's a full-time member of the lab who just does bioinformatics analysis all the time and mentors other people about it. And then I also have a short-term postdoc who was here over the summer, basically finished his PhD, wanted to learn a little bit more bioinformatics and a little bit of immunotherapy in terms of the biology concepts. And he's getting ready to launch into his own position in biotech. So I currently have 3 people in the lab. We're gearing up to have undergrads and graduate students starting in the fall.
I guess it's a good time to ask if any listener in the area if you're actively still looking for members of your lab.
Absolutely. People can reach out anytime. If you have interest in immunotherapy or immunotherapy for solid tumors specifically, please let me know. We're always looking for really motivated and enthusiastic researchers.
All right. Well, at the end of the show, we like to ask a question not directly related to research or careers. We call it the fun question, but it's usually a very insightful one. So if you were not a scientist, what would you be instead?
I love this question. If I were not a scientist, I would be a scuba dive instructor. We went actually to a scientific conference when I was in graduate school out in Australia and took a few extra days, and we went up to the Great Barrier Reef and did a live aboard scuba dive experience. And so I got to learn how to scuba dive. And we went down for a training dive and we came back up, and the guide tapped on my regulator and said, "Did you even breathe down there? You barely used any oxygen." So that was a really big indicator just how comfortable I was under the water. I was a swimmer in high school, loved the beach and snorkeling all the time when I was growing up. And more recently, post-pandemic, when we had a little bit of vacation money to use, we went to Tahiti and we went scuba diving at several different locations in the Polynesian Islands. And I could live there. I just loved being underwater. I loved the ocean life. It's like another world.
From now on, every holiday is a scuba diving destination.
As many as I can deliberately get everyone to go to a scuba dive location, I absolutely try to manipulate vacations that way.
Excellent. That sounds like a great plan. It's been so nice talking to you. It's a really great conversation. I mean, I could speak about T-cell therapy all day long, but we can't. We do have allocated time for this. But as you mentioned, any listener interested for a position, reach out. New lab, new PI. This is a perfect situation, perfect storm for a successful graduate or postgraduate experience. So all the best. Congratulations, and good luck in the years to come.
Thank you so much. This was really a pleasure. I'm really honored to be here. So thank you so much for the invitation.
Pleasure. Thank you.
Well, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on Twitter at Immunopodcast or via email at info@immunologypodcast.com if you have any feedback or you would like to suggest a guest. See you next time.